Welcome back, focused learners. Let's jump right into today's explainer because we are looking at a biological molecule that is honestly incredibly misunderstood. I mean, we hear its name and we immediately think of danger, right? But today we're actually going to look at the biochemistry. We're going to map out exactly how this molecule works, how our bodies literally build it from scratch, and the really fascinating pharmacology we use to keep it in check when things go wrong. Let's get into it. So, is cholesterol actually the bad guy? It's a question we've got to ask right out of the gate. Because for decades, it's been painted as this ultimate villain, the dietary demon responsible for clogged arteries and heart disease. But when you look at the actual biology, you quickly realize something crazy. If you were to completely remove cholesterol from your body right now, you wouldn't just be unhealthy. Your cells would literally fall apart. So we really need to completely reframe how we view this molecule. Here is our roadmap for today. First, vital roles in the body. Second, how cholesterol is made. Third, the lipoprotein transporters. Fourth, first-line cholesterol drugs. And finally, advanced lipid therapies. Starting with Section 1, vital roles in the body. The hero we actually need. Because before we can even talk about how to lower it, we have to understand why our bodies are so fiercely protective of it in the first place. Think about this concept of a fluidity buffer. Your cell membranes, they aren't rigid walls, right? They're these fluid, dynamic structures made of phospholipids. And cholesterol literally embeds itself right inside that phospholipid layer. It acts almost like a microscopic thermostat, or like antifreeze for your cells. At low temperatures, when the membrane wants to pack tightly and freeze solid, cholesterol wedges in there to keep things fluid. And then at high temperatures, when the membrane threatens to get way too loose and just melt apart, cholesterol restricts that movement to stabilize it. It really is the ultimate biological stabilizer. And that stabilizing effect is absolutely crucial for ATP synthesis. It actually reduces the passive leakiness of the membrane to hydrogen and sodium ions. Now, because oxidative phosphorylation relies entirely on really precise proton gradients, without cholesterol sealing up those membranes, our cells literally couldn't generate energy effectively. On top of that, it organizes these specialized microdomains we call lipid rafts. You can think of them like tiny little landing pads that facilitate lightning-fast cell signaling and perfectly timed endocytosis. But wait, the job description doesn't stop there. Cholesterol is the starting substrate for all of your steroid hormones. We're talking corticosteroids and your sex hormones. When you're exposed to UV light, it transforms into vitamin D3. Over in the nervous system, it's a major structural component of the myelin sheath that insulates our nerve fibers. In fact, a massive 20% of your body's entire supply is sitting up in your brain, balancing synaptic fluidity so vesicles can actually fuse and release neurotransmitters. Oh, and in your skin, it combines with ceramides to form a lipid mortar that basically keeps your body's water from evaporating. Moving to Section 2, how cholesterol is made, the cellular factory. Because it's so incredibly vital, our bodies don't just leave this up to our diet. We have a massive cellular factory, mostly in the liver, dedicated to manufacturing this stuff from scratch. And this biosynthesis is a seriously complex, energy-intensive assembly line. It kicks off with basic glucose breaking down into acetyl-CoA. That condenses into citrate, which moves out to the cytoplasm to form H&G-CoA. Then, that gets reduced to mevalonate, which goes through this really long series of transformations, hitting squalene and manostrol along the way until it finally becomes cholesterol. This immense investment of cellular energy just proves how absolutely essential the final product is for our survival. Now, within this whole pathway, there are two specific factory managers you need to know about. First, we have the ATP citrate lyase enzyme, which catalyzes a key step right in the cytoplasm. And second, we have HMG-CoA reductase. This is the big one. This is the primary rate-limiting step of the entire operation. These two enzymes completely dictate the speed of the whole factory. Keep these in the back of your mind, because they are going to become absolutely crucial targets when we get into the pharmacology later. Which brings us to Section 3. The lipoprotein transporters, delivery and cleanup. Remember, cholesterol is a lipid. It's a fat, so it cannot dissolve in our watery blood. It needs specialized transport vehicles to get around. You've definitely heard of these vehicles referred to as good and bad cholesterol, but really it's way more accurate to just think of them as different delivery trucks. LDL, or low-density lipoprotein, is your outbound delivery system. It binds to receptors on peripheral tissues like your muscles and adrenal glands, dropping off cholesterol for cell membranes and hormone synthesis. The danger only kicks in when there's just way too much LDL in the blood. It starts depositing that cholesterol into your arterial walls, which is what drives atherosclerosis. On the flip side, HDL, or high-density lipoprotein, is the cleanup crew. It performs reverse cholesterol transport. It literally vacuums up the excess cholesterol from peripheral cells, like the macrophages in those arterial walls, and carries it straight back to the liver to be cleared out. Section 4. First-line cholesterol drugs. Taming the system. This is the rehabilitation phase, where pharmacological marvels step in, because that inbound and outbound transport system has fallen dangerously out of balance. The absolute go-to first-line therapy for most hyperlipidemias is a massive class of drugs called statins. We're talking about medications like atorvastatin, rosuvastatin, and simvastatin. And hey, remember that big factory manager we talked about earlier, HMG-CoA reductase? Well, statins act as a perfectly targeted tactical strike. They directly inhibit that specific enzyme, effectively shutting down the liver's entire cholesterol manufacturing plant. But here is where the biology gets truly beautiful. Let's trace this. It's called the SREBP pathway. When statins inhibit that enzyme, the cholesterol levels inside the liver cells, the hepatocytes, naturally begin to drop, and the liver panics. To survive, it sends these special proteins called SREBPs straight into the cell's nucleus to activate genes that build more LDL receptors. The liver pushes all these brand new receptors out to its surface, and they act like giant magnets, physically pulling circulating LDL right out of the bloodstream to harvest its cholesterol. So by shutting down internal production, the drug actually tricks the liver into cleaning the blood for us. It is just a brilliant physiological feedback loop. And now, Section 5, Advanced Lipid Therapies, the backup arsenal. Because as amazing as statins are, they aren't perfect. Some patients get statin-associated muscle symptoms, or they have severe genetic conditions where statins alone just aren't going to cut it. What's so fascinating about these advanced backup therapies is how they strategically attack completely different parts of the cholesterol supply chain. For example, bempedoic acid goes after that other factory manager we mentioned earlier, the ACL enzyme. Meanwhile, PCSK9 therapies focus on protecting your LDL receptors. Other drugs work entirely down in the gut. Ezetimibe blocks absorption directly, and bile acid sequestrants literally trap cholesterol byproducts. And finally, you have fibrates, which specifically target lowering triglycerides. Let's look a little closer at how a couple of these actually operate. When you really dive into these mechanisms, the sheer precision is mind-blowing. Take ezetimibe. It acts like a bouncer at a club. It selectively inhibits the NPC1L1 transport protein right inside the small intestine, essentially blocking the main port of entry so dietary cholesterol can't even get into your body. Then we have the PCSK9 therapies. Now, normally, this protein called PCSK9 wanders around your blood marking your helpful LDL receptors for destruction. By using monoclonal antibodies to neutralize PCSK9, we're basically deploying molecular bodyguards. They save our LDL receptors from being destroyed so those receptors can keep actively pulling bad cholesterol out of the blood. Okay, and this number right here, 95%, this is the key to yet another brilliant therapy. Normally, your liver converts some of its cholesterol into bile acids to help you digest fats in your diet. And incredibly, 95% of those bile acids are efficiently reabsorbed back into the intestine and recycled. Bile acid sequestrants, like cholestyramine, are these non-absorbable resins that physically bind to the bile acids in your gut, forcing your body to just excrete them. Because that recycling loop is now broken, the liver is forced to burn through its own internal cholesterol reserves to synthesize brand new bile acids, and that drastically lowers the body's overall cholesterol burden. So as we wrap this explainer up, I really hope you can see how incredible this whole narrative arc is. We've traced cholesterol all the way from its status as a critical, life-sustaining hero. I mean, a molecule without which our cells would literally melt and our skin would leak, to becoming a rogue villain when its transport mechanisms go awry. And finally, we've seen how modern pharmacology elegantly manipulates the body's own feedback loops to bring it all back into perfect balance. Which leaves us with a final thought to ponder today. Biology rarely makes mistakes. If our bodies have evolved entire complex molecular assembly lines, specialized delivery trucks, and crazy efficient recycling systems just to build and maintain cholesterol, what other so-called bad molecules in our diets or bodies are actually just misunderstood masterpieces waiting for us to understand their true purpose?